Imagine for a second that you're trying to understand how a massive, bustling city works, right? Like, you want to know everything about the economy, the traffic, the people, but there's a catch. You're only allowed to look at one single building at a time.
Which is basically an impossible task.
Completely impossible.
Yeah.
But honestly, for over a century, that is exactly how biology has treated the mammalian body, especially the laboratory mouse. We'd analyze a piece of the liver today, maybe some brain tissue tomorrow, but we were totally blind to the big picture. We couldn't see the whole city at once.
Right, it was all incredibly isolated.
Yeah. But today, we're doing a deep dive into a paper that basically changes everything. It's from the journal Cell, published in 2026 by Clevenger and colleagues, and they essentially created a Google Earth for the entire mammalian body.
It really is a massive paradigm shift. I mean, the stated mission of this team was wild. They set out to map every single cell type, every organ, and the entire simultaneous gene expression profile across the whole body of a mouse, all at the exact same time.
Which just sounds like science fiction. So we have this incredible source material, and we're going to unpack exactly how they did it, what they found, and why this fundamentally rewrites our understanding of disease.
Yeah, and the term we need to define right up front here is spatiotranscriptomics. It's a bit of a mouthful, but it's the core of everything they did.
Spatiotranscriptomics. Okay, break that down for us.
Sure. So transcriptomics is just the study of RNA, right? The transcripts. It tells us what specific genes are actively turned on in a cell at any given moment. But traditionally, to get that RNA, we had to do what's called bulk sequencing. We'd take a chunk of tissue and basically blend it up into a cellular smoothie.
A smoothie, right. So you have strawberries and bananas in there, but you have literally no idea where they were sitting in the original fruit bowl.
Exactly. You lose all the geography. And even when we got better at this, with single-cell sequencing, where we could look at individual fruits from that smoothie, we still didn't know their original zip code in the body. So the spatio part of spatiotranscriptomics fixes that. It means mapping not just what the genes are doing, but exactly where they are doing it in physical space.
So imagine if you could have a map of your own body like that right now. Every single cell's activity, perfectly mapped in 3D. It's mind-boggling. But wait, how do they actually pull this off physically? Because you can't just put a whole mouse into a deli slicer. I mean, if you slice an animal thin enough to see individual cells, wouldn't it just turn into a puddle of mush?
You'd think so, yeah. The mechanics are actually fascinating. They use this highly specialized tool called a cryomacrotome, and it relies heavily on flash freezing. They take an adult lab mouse, specifically a C57B-L6J, which is, the standard genetically identical workhorse mouse used in labs everywhere.
Right, a classic lab mouse.
Yeah. And they freeze it so incredibly fast in liquid nitrogen that the water inside the cells doesn't have time to form ice crystals. It turns directly into glass. It's a process called vitrification.
Oh, wow. Wait, why is the ice crystal part so important to avoid?
Because if water freezes normally, it expands. Ice crystals would act like microscopic daggers and completely shred the delicate cell membranes.
Okay. So by turning it to glass, everything is perfectly locked in place without bursting.
Precisely. Once it's basically a solid block of glass, this cryomacrotome uses a temperature-controlled, ultra-sharp blade to literally shave off slices of the entire whole-body mouse. And these slices are just 10 micrometers thick.
10 micrometers, just to give you guys some context. A single strand of human hair is like, 70 to 100 micrometers thick?
Yes, something around there.
Yeah. So they were slicing a whole mammal lengthwise into ribbons that are a fraction the thickness of a hair. And because it's vitrified glass, it all stays intact. The paper notes they perfectly preserve things like the fragile intestinal mucosa, the soft bone marrow, even the delicate internal structure of the eye.
It's incredible engineering. So they take that ultra-thin slice and put it on a slide. Now, before they get to the genetics, they need a regular visual map. They do this using H&E staining, which has been the gold standard in pathology forever.
H&E, hematoxylin, and eosin. Actually, when I was trying to wrap my head around this, I found a great little mnemonic for the visual memory of it. Think of the H, hematoxylin, as highlighting the heavy blueprints. It dyes the acidic DNA in the cell nucleus dark purple. And the E, eosin, paints the environment. It dyes the surrounding cell cytoplasm bright pink. So purple blueprints, pink environment.
That is a brilliant way to remember it. The heavy blueprints and the environment. So, okay, they have this beautiful pink and purple map. But here is where the magic of the Array-seq platform comes in. The glass slide they use isn't just a piece of glass. It is printed with millions of microscopic chemical barcodes.
Oh, so the slide itself is smart.
Very smart. They take the tissue on the slide and they basically permeabilize it. They chemically poke tiny holes in the cell membranes. This causes the active RNA inside the cells to basically drop straight down onto the glass.
And since it drops onto the barcodes.
Exactly. When the RNA hits the glass, it binds to a specific barcode. So later, when they sequence all that RNA, they can look at a piece of data and say, oh, this barcode tells me this exact gene was active at coordinate XY on the slide.
That is wild. The RNA literally gets stamped with a spatial zip code.
Yep. And through this, they captured the genetic data of 16 distinct organ systems. And what's so cool is how perfectly the invisible gene activity matched the physical anatomy we all know. Like, in the stomach, the genetic data easily told apart the parietal cells, which make stomach acid, from the chief cells, which make digestive enzymes.
Right, because their gene activity is totally different, even if they're right next to each other.
Exactly. They even tracked the whole path of digestion in the small intestine just by looking at the active genes. It went perfectly from proximal markers like DE430 and OAT right down to the distal marker KCAR20.
Okay, let's do a quick recap of this map-making phase before we move on. Point 1, they flash freeze the mouse to glass so it doesn't turn to mush. Point 2, they slice it 10 microns thin. Point 3, they dye it pink and purple to see the geography. And point 4, they use bar-coded slides to grab the genetic zip code for every single spot. It's a flawless system.
It really is.
It makes you wonder, if you could take a 10-micron slice of any mystery in your own body right now, what organ would you want to look at first? I'd probably want to see my brain, just to figure out what genes are misfiring when I walk into a room and instantly forget why I'm there.
I think we'd all like to see that map. But, having all these genetic zip codes actually creates a whole new problem. You have the geography, sure, but a map is kind of useless without a legend.
Right, a zip code isn't a person. You know the genes, but who are the actual citizens living in these tissues?
Exactly. Just knowing a gene is active doesn't definitively tell you what specific type of cell you're looking at. So to build the roster of citizens, they had to bring in massive computational power. They used a database called CellKB. They pooled together 466 different single-cell RNA studies from the past, which gave them this insane master reference data set of 59 million single cells.
Wait, let me make sure I'm grasping the scale of this. The actual slice of the mouse they're looking at has, maybe 5 million nuclei on it?
Roughly.
So you're saying the algorithm takes the RNA profile of one tiny dot on the slide, and it checks it against a master database of 59 million molecular mugshots just to find a match.
That is exactly what it does. It's this colossal, pattern recognition task. And by comparing their slide to those 59 million mugshots, they defined 495 highly specific granular cell types. And they successfully mapped 379 of them across the whole mouse body.
And the demographics of that cell census are actually super surprising. When you map the whole body, skeletal muscle cells are just incredibly dominant. They covered up to 44% of all the mapped spots.
Which makes sense structurally, but it's still jarring to see on paper.
And the heart. The heart was basically a monoculture. It was like 95% cardiac muscle. But I think the coolest part was how they found the microscopic stuff hiding inside the big organs.
Oh, absolutely. The resolution was stunning. Take the pancreas, for instance. They didn't just see a pancreas blob. They pinpointed the tiny pancreatic islets with amazing detail.
Ugh. The islets, the little hormone factories. Okay, let's bring back a classic, if slightly ridiculous, medical mnemonic for the pancreas cells. Alpha cells make glucagon, beta cells make insulin, delta cells make somatostatin. The letters are AGBIDS. So always gluttonous, big insulin, don't stop.
I mean, whatever helps it stick in your brain. But yes, they literally saw that play out at the genetic level. They saw the GAT gene lighting up the alpha cells, the NS2 gene in the beta cells, and the SUSA gene in the delta cells. They even found these tiny specialized glands, like the Harderian gland in the eye, lighting up with the urod gene.
Okay, so quick recap for this cell census segment. Point 1, they built a database of 59 million single-cell mugshots. Point 2, they matched their slide's RNA against that database. Point 3, they successfully ID'd 379 distinct cell types right down to microscopic hormone factories.
And mapped them all spatially.
Right. And, knowing that almost half your body mass is just purely skeletal muscle, it really shifts how you view your daily metabolism, doesn't it? Like every time you eat, you're mostly just feeding this massive muscular engine.
It definitely puts things in perspective. But as amazing as this is, there is a giant elephant in the room here.
The cost.
The cost and the computational weight. Array-seq is incredible, but actually sequencing all that RNA is notoriously expensive. It takes pricey reagents, massive server racks, days of processing time.
Right, you can't exactly do this for every single lab mouse, let alone roll it out for routine human biopsies. There has to be a shortcut.
And the researchers knew that, so they developed an artificial intelligence solution, a machine learning pipeline called Label. The entire goal of Label is to look at a standard, cheap H&E image, just those basic pink and purple shapes, and accurately guess the cell types and the active genes without actually doing any of the expensive RNA chemistry.
Okay, that sounds like literal magic. How can an AI look at a pink and purple texture and know what invisible genes are turned on inside the cell?
Well, it relies on two algorithms working together. The first is a pathology foundation model called UNI. UNI has been pre-trained on millions of medical images, so it essentially speaks the visual language of biology. It knows exactly what a nucleus, a membrane, or a tissue fold looks like.
Okay, so it has good eyes.
Right. But the real brain of the operation is the second algorithm, the spatially aware K-nearest neighbors classifier, or KNN for short.
K-nearest neighbors. Okay, let me try an analogy to explain this one. Imagine you're looking at a satellite photo of a mystery person standing in a city. You can't see their face, but you look at the buildings around them. If they're surrounded by classrooms, a playground, and a school cafeteria, your brain naturally guesses, oh, this person is probably a teacher. Is Label doing that with cells?
That is a brilliant analogy, yes. But let's refine it just a little bit for the biology. The K in KNN just stands for a variable number. So the AI isn't just looking at the general neighborhood. It looks specifically at the other cells physically touching the mystery cell.
Ah, I see.
So if it looks at its 10 nearest neighbors, and they're all structurally arranged like bile ducts and portal veins, the algorithm calculates a very high probability that the mystery cell is a hepatocyte, a liver cell.
So it's heavily relying on spatial context clues. And does it actually work?
Extremely well. When they scored its accuracy, where 1.0 is absolute perfection, Label identified hepatocytes with a score of 0.98. It was incredibly accurate with renal cells in the kidney, too.
But it wasn't totally flawless, right? The paper mentioned a pretty realistic limitation. Because the AI is relying entirely on visual shapes and neighborhood context, it can get fooled by convergent evolution.
Right, the fact that nature tends to reuse good structural designs. So without the RNA chemistry to double-check, the AI sometimes got confused between tissues that look like identical twins but act totally differently.
Like mixing up cardiac tissue with skeletal muscle? Because they're both dense, striated muscle fibers?
Exactly. Or it occasionally mistook salivary glands for kidney tissue because they both have these complex networks of secretory ducts.
Okay, time for our AI recap. Point 1, sequencing RNA is too expensive for everyday use. Point 2, they trained an AI called Label to just look at the cheap pink and purple slides. Point 3, the AI uses nearest neighbor context clues to guess the cell type with high accuracy, though it sometimes gets tricked by look-alike tissues.
That sums it up perfectly.
And imagine the time saved in pathology labs if this scales up. If an AI can virtually stain a human biopsy and map the genes instantly, you could get complex medical test results back in minutes instead of waiting agonizing weeks.
The clinical applications are just staggering to think about.
But you know, a static map, even a brilliant AI-generated one, is just a snapshot. And life isn't static. So the researchers decided they needed to take this brand new whole-body map out for a test drive. What happens when disaster strikes a metropolis?
And this is where the technology really shines. They used a well-established clinical model of systemic inflammation called endotoxemia. Essentially, they injected the mice with LPS, which stands for lipopolysaccharide.
And LPS is basically a toxin from bacteria.
Right. Right. It's a molecule found on the outer membrane of certain bacteria. When a mammal's immune system detects LPS floating in the blood, it completely panics. It thinks there is a massive, life-threatening bacterial invasion, and it triggers a severe systemic inflammatory response across the whole body.
So they basically artificially induced a massive full-body infection just to see what the map would do. Now, I would assume the map just lights up with immune cells rushing around the blood vessels.
Well, the immune cells absolutely react, but that wasn't the big shocker. Because they were looking at the entire body at once, they watched organism-wide changes happen in real time. They saw a massive activation of defense genes controlled by these two master switch proteins, STAT1 and IRF1.
STAT1 and IRF1.
Yeah, they are transcription factors. They literally enter the cell nucleus and flip the on switch for immune defense programs. But the crazy part was where these switches were being flipped. It wasn't just in the hematopoietic cells, the blood and immune cells. These defense programs were activating in the non-hematopoietic cells.
Wait, really? You mean the physical structural cells of the organs were joining the fight?
Yes. The cells that just build the organs were actively turning on immune defenses.
That is wild. It's like sounding a fire alarm in a giant skyscraper, and you're expecting the firefighters to show up. But suddenly you realize the accountants, the janitors, and the baristas in the lobby are all dropping what they're doing and grabbing buckets of water. The entire body becomes one unified immune organ.
That's a great way to visualize it. And they proved it physically, too. They stained the tissue for a structural protein called vimentin. Vimentin is crucial for cells like fibroblasts and stellate cells, which act as the scaffolding of your organs.
Okay, the scaffolding cells.
Right. And during the simulated infection, they saw a massive increase in these fibroblasts in the heart, the liver, the intestines, the skin, everywhere. When these structural cells hear the STAT1 and IRF1 alarms, they multiply and physically remodel the tissue. They might lay down extra collagen to stiffen the area, basically building walls to trap the infection and help the immune cells move around.
They actually shapeshift the organs. That completely changes how I view getting sick. Let's do a quick recap of this stress test. Point 1, they injected a bacterial toxin to cause full-body panic. Point 2, master switches called STAT1 and IRF1 activated everywhere. Point 3, the basic structural cells of the body actually remodeled themselves to fight the infection.
Gone.
So, question for you to ponder. If every single non-immune cell in your body plays a role in fighting an infection, shouldn't that completely change how we design drugs? Like maybe antibiotics shouldn't just target the bug or the immune system. Maybe we should be designing drugs that help the baristas and janitors, the structural cells, fight the good fight.
It definitely opens up totally new avenues for therapeutics. But, as with all groundbreaking science, we have to stay grounded about where this currently sits. It's a monumental proof of concept, but it's not quite the finish line.
Yeah, let's talk about the limitations, because it's not perfect yet. You mentioned that cryomacrotome earlier. I'm guessing my local urgent care doesn't have one of those in the back room.
Definitely not. The physical sectioning of whole mammals remains incredibly difficult, specialized, and expensive. But there's also a big data limitation. The AI model, Label, was trained on a single sagittal plane. So just a 2D side view slice of female mice of a single specific age from a single uniform genetic background.
Right, the lab standard C57BL mouse.
Exactly. But biology in the real world is messy and heterogeneous. To be a truly universal tool, this whole framework needs to be expanded into 3 dimensions. And the AI needs to be trained on mice of different ages, both sexes, and all sorts of different genetic conditions. A map of a healthy young female mouse isn't going to perfectly predict the tissue of an elderly male mouse with, say, diabetes.
That makes total sense. The map has to represent the actual population.
Yeah.
But even with those limitations, the big picture here feels huge, especially for things like drug toxicology testing.
Oh, absolutely. Right now, if a pharma company makes a new drug targeting the liver, they mostly just check the liver. But they might completely miss the subtle collateral genetic damage the drug is causing in the stomach lining or the bone marrow.
But with this Google Earth map, you could dose a mouse and instantly see the ripple effect across all 379 cell types at once.
Yes. And it also changes how we map genetic mutations. If a patient has a single mutated gene, we won't have to guess which organ systems are affected. We can literally see the entire systemic web of consequences.
And that actually leads me to a final sort of provocative thought I want to leave you with today. If we keep scaling this up, and eventually we're going to map the entire human body at this incredible spatial and genetic resolution, how are we going to redefine what a disease actually is?
It's a deep question.
Right. Because if we can literally see that a tiny change in your gut instantly remodels the physical scaffolding of your heart, are we going to have to stop using terms like isolated liver disease or isolated heart disease? Will we eventually realize that literally every illness is a systemic illness?
The human body really is just a single, deeply interconnected ecosystem. When one neighborhood struggles, the whole metropolis adapts to carry the load.
Think about that the next time you feel a little under the weather. You aren't just a collection of parts sitting in a bag. You are a bustling, billion-strong metropolis. And right down to the microscopic level, every single citizen is playing their part. Thanks for taking this deep dive with us.